Today we'll be looking at how the promise of plants for engineering human tissue is allowing us to grow ears from apples. If you're new to this channel, welcome. This is Mr. Singularity, where we explore the illuminating scientific and technological breakthroughs shaping the future as we know it. It seems that inspiration for game-changing research will come from everywhere. The first antibiotic, penicillin, was given to us by a moldy bacterial plate. The zapping of yeast with platinum electrodes led to a potent chemotherapy drug, cisplatin. For Dr. Andrew Pelling at the University of Ottawa, his radical idea originated from a science fiction cult classic called The Little Shop of Horrors. Specifically, he was fascinated by the film's main antagonist, the man-eating plant named Aubrey II. What you have here is a plant-like creature with mammalian features, so we started to wonder, can we grow this in the lab? The ultimate goal of Pelling, of course, is not to bring a science fiction creature to existence. Rather, he wanted to see if grocery store purchased plants could provide the requisite structure for the engineering of human tissue replacement. The rise of mechanbiology. Growing the human ear out of apples might seem irrational, but Pelling's main insight is that the fibrous interior of the apple is remarkably similar to the microenvironments widely used in laboratories for bioengineering human tissue. For example, scientists typically carve or print 3D hollow support structures out of expensive biocompatible materials to create a replacement ear. Then they seed human stem cells into the structure and carefully supply a cocktail of growth factors and nutrients to enable cells to expand. After weeks and months of incubation, the cells spread and differentiate into skin-like cells on the scaffold. The effect is a bioengineered replacement ear. The problem, The incredibly high entry bar, stem cells, growth factors, and scaffolding materials are both difficult and costly to acquire. But are these main components really necessary? We often think of biology through genome lenses or biochemistry, but cells and tissues are living components. They stretch, compress, and shear, creating mechanical forces that act on each other. In a number of studies, Pelling and others have found that these mechanical forces are not just a side product of biology, rather they seem to be essential in controlling the fundamental molecular machinery of the cell. Another early study found that every stage of embryo growth, a fundamental process in biology, can be regulated and controlled by mechanical knowledge. In other words, physical forces will drive cells to divide and migrate through tissues, while our genetic code directs the development of the whole body. In the lab, stretching and mechanically stimulating cells often seem to radically alter their behavior. In a single assay, Pelling's team peppered cancer cells on a sheet of skin cells grown at the bottom of a petri dish. The cancer cells huddled together in tiny balls, creating a distinct barrier between the microtumor and the skin cells. But when the team placed the entire cellular system in a device that extends it minutely, mimicking the body's breathing and movement, the tumor cells become vicious, tunneling through layers of skin cells. There is no gene alteration or biochemistry going on here. This is purely a mechanical effect. There is a fundamental link between these things. Even cooler, active movement is not required to transform the actions of cells through mechanical forces. The shape of their microenvironment is enough to direct their behavior. For example, when Pelling placed two types of cells into a physical structure with grooves, the cells were self-segregated within hours, with one type growing in the troughs and the other on the upper ledges. By simply sensing the form of the grooved surface, they learned to differentiate and spatially pattern over long distances. Takeaway: using shape alone, it is possible to induce cells to form complex three-dimensional patterns. This is where the apple comes in. Apple on my neck, Under the microscope, the microenvironment of the apple is the same length scale as the surfaces built for the manufacture of replacement tissues. The discovery led the team to wonder, is it possible to make use of the surface pattern of plants to grow human organs? They took an apple and washed away all of its plant cells, DNA, and other biomolecules to test it. This left them with a fibrous scaffold, things that are usually stuck in the teeth. When the team stuck human and animal cells inside, the cells started to expand and spread. Encouraged, the team hand-carved the apple into the form of a human ear and repeated the above procedure. Within weeks, the cells invaded, turning the apple chunk into a fleshy human ear. Of course, it's not enough to have the right form. With the body, the replacement tissue must also survive. Next, the team inserted an apple-based scaffold directly under the skin of the mouse. Not only did the mouse's healthy cells invade the matrix in just 8 weeks, but the rodent's body also developed new collagen and blood vessels that helped keep the scaffold alive and healthy. It has 3 important aspects for engineered tissue. It is healthy, biocompatible, and comes from a renewable, ethical source. This thing is becoming a living part of the body, and it used to be an apple, and we did this by going to the grocery store. Moving to the hospital room. Pelling is particularly excited by its simplicity. It does not require stem cells or exotic growth factors to function. The elegant approach takes advantage of the physical structure of the plant. The team is now expanding its work to 3 major fields of tissue engineering: soft tissue cartilage, bone, spinal cord, and nerve repair. The trick is to adapt the particular microstructure of the plant to that of the tissue. It's really exciting to see these kinds of wild ideas translate like this. And why do we limit ourselves to the body parts that nature has given us? If the shape of a scaffold is the sole detriment of tissue or organ engineering, why not make it our own? Pelling took the concept and ran with it, commissioning a design firm to draw out the scaffold for 3 distinct types of ears: the normal human ear, the pointed Spock-shaped one, and the waved one designed to block or improve different frequencies to theoretically augment hearing. The point I want to stress is the strength of the blue sky way of thinking is, in fact, to combine it with the rigor of the scientific method. At the end of the day, this is how we can build more inventions and solve problems. What's your take on this? Let me know down in the comments below and check out one of these other videos. This has been Mr. Singularity, and I'll see you on the next one.